to the last Science Cafe that we're having this summer. The next one is in 3 weeks, so keep that on your calendar. But today, so I'm Dustin Updike. I'm an assistant professor over at the MDI Biolab. And today we have with us David Fay. David is coming to us from the University of Wyoming, but he is a Northeasterner. He grew up in Vermont area. He did his PhD in molecular biophysics and biochemistry at Yale. Went to Tufts for undergraduate school. And then he did his postdoc at University of Colorado in Boulder. And it's there where he started using C. elegans, these small little worms that he's going to tell you about, to study some biological questions. So David's now in Wyoming, and Wyoming's my home state. That's where I went. I grew up. And about 2001, about the time that I was leaving Wyoming to go to graduate school, David came to the University of Wyoming as an assistant professor. And so our paths didn't cross for a couple of years, but I knew about David because when I started graduate school, I started working with these little worms as well. And there was this guidebook on worm biology that I relied on heavily, and it was written by David. It was called Worm Breeding for Dummies. And I think that after that you changed the title to Worm Breeding for Super Geniuses.
Yes.
So that you didn't get sued or something like
that.
Yeah,
that's right.
So and this resource is something that every time I get a new person in my lab, I give him David's book because he wrote the book on worm genetics, and so I am pleased to have him with us today. Let's see. So he's now a full professor at the University of Wyoming, and he runs a successful research program and has been doing that for the past 13 years. He's authored a few dozen fantastic publications, and he serves as editor on several journals. And is currently the director of the Molecular and Cellular Life Sciences program there at the University of Wyoming. And thank you. I'm glad to have you today, and I'll turn it over.
Thank you so much. See, now, the science crowd never applauds until the end because they're reserving judgment, just so you know. So it's great to be here. Dustin's actually kind of a legendary undergraduate from my department because I started to hear about him the moment I walked in, and so it was great when Dustin and I ran into each other at meetings. And it's just really it's an honor to be here, and I can relate to this place a lot because Wyoming's a really beautiful place too, and there's not a lot of people don't get this, but you can kind of have your cake and eat it sometimes in life. You can do science and do really good work in an absolutely gorgeous setting with a fairly laid-back lifestyle otherwise. So this place is actually a lot closer to home than you'd think a landlocked state that gets 9 inches of precipitation a year might be. Okay, so this first slide just sort of launches right into it. This is worm embryogenesis, and what you're watching is you're watching cells divide, and at some point I'll stop this and point out something to you. Let's see. It's just in a little bit. I'm going to point out something that looks like almost nothing. Okay, I'm going to point these out. So you see this kind of little circular button-like shape there and there? They're a little bit hard, but they look slightly different. These other things that are nuclei look sort of like craters, where these look, at least to my eye, like little blips or hills. And if we kind of keep running this, you'll see this happens quite frequently. I'll stop it in 1 or 2 other places where there's sometimes, whoops, now it's starting to move around too much. We can't see anything. And I've somehow my pointer didn't. Okay, so there's these little blips, and what happens is they appear and they disappear. Doesn't seem like much, but I'll get to why that's important. I'll start out not by talking about my work. I'll say something just a little bit about that at the end. But I feel like this talk is more general, and what I want to convey is that you can't anticipate where the great discoveries are going to be, and there's a lot of good reasons to work on simple, humble organisms like the nematode. So this is what it grows up to be. This is not a night crawler, so these are these are tiny little creatures. They're about 1 millimeter long. They live in the soil. They're free living. They're non-parasitic, although they have relatives that are parasitic, but somewhat distant. So there's nothing to fear from them. And if you actually dig up a clump of soil in your backyard, I guarantee you've dug up probably thousands of these guys. So this is this is what they look like when they crawl along. And this is bee crawling along on a petri dish. They just kind of slither along. They're kind of hypnotic. They're nice to watch. That's its tail. The head will probably, if this is looped, I guess it's not, the head will come up. One of the things that's kind of neat about it is that it's totally transparent. And the number of, you can see every cell in the organism with a microscope, so there's no skin or pigmentation that you have to worry about. They live in the soil under, they don't get exposed to light. So they haven't bothered with pigment. This thing right here, you can see like, it looks like a row of bricks almost, with a little circle in the middle. This is actually a nucleus, and this is an oocyte. And just in about 3 days, it will go from an egg, and you can actually see this is a hermaphrodite worm. So it produces its own sperm and eggs. It self-fertilizes, and it produces these embryos, these sort of circular oval-shaped things inside it. And that's what we were looking at earlier. We were looking at divisions of cells within the embryo in the previous movie. And so they'll lay an egg, and about 3.5 days later, it's a worm that's laying its own eggs. So incredibly rapid generation time, and each worm will give rise to about 300 progeny. So, yes, their function, that's a great question. I think they're cleaner upper, so I don't know a lot about the ecology of worms, of these worms. I think they live in the soil and they eat things like bacteria. They're part of some life cycle, so they eat things like bacteria and yeast. I think they grow, they get transported around apparently in snails. Wasn't that one of the theories? There's some people, so most of the people, confession here, I'm not a nematologist, so most of the people who work in C. elegans couldn't tell you much of anything about their normal function or what they do out there in the wild, which is not an insignificant question. We use them sort of as little laboratory living test tubes to sort of get at molecular genetic underpinnings of biology, you could say. But that's a good question. And please stop to ask questions.
Do they freeze?
What they do is they do freeze worms. So I don't know how well in the wild worms recover from freeze-thaw cycles, but they're one of the few organisms that are multicellular metazoa organisms that you can put in a test tube and throw in the -70 or in liquid nitrogen essentially and forget about them. Now we add some things like glycerol and a few things that kind of apparently help stabilize their state in the frozen so they don't get big ice crystals in their cells while they're freezing. But yes, they're cryogenic. You can freeze them, which is really nice. So in flies, sort of one of our competing organisms, and we don't like to badmouth them, but because they have a lot of, there's great strengths to the fruit fly, but you can't freeze them. And so anytime you make a strain, you're saddled with that strain for life. And we make hundreds or thousands of strains, and it's nice to be able to throw them in the freezer when we're not working with them. And it's a good resource for the field too. Yeah, good question. Okay, so this is one of the interesting things about C. elegans, and is one of the strengths of the system as well. They have this so-called hardwired developmental lineage. What that means is if you watch one embryo and you've recorded it and you understand what's going on, if you've seen one, you've seen them all. It's really true for worms. So the cells divide at the same time, in the same orientations, and then they ultimately differentiate into the same cell types in the exact same reproducible pattern from one C. elegans embryo to another. So the idea would be, these are really hard to see here, so I've blown it up here. The idea is you'd have a mother cell somewhere in here, that's a vertical line, that would be A. It would give rise to 2 daughter cells, B1 and B2, which would give rise to maybe these 4 daughter cells, etc. And there's a guy named John Sulston who in England mapped this out. He had a good mind for this sort of thing. Now it would be fairly trivial because you could do imaging and 4D analysis, what they call it, and whatnot. He sat at a microscope for 5 years and just drew and just kind of had it all in his mind and sketched it out. And that was really one of the things that established the field. So again, all these divisions are taking place. Now the next slide traces the number of cells over time in an embryo. Okay, so we're looking at the number of nuclei and sort of going down here as minutes, and then sketched out here are some of the different developmental stages. What I want to point out is the obvious. You start out with one cell, and you divide and you get more cells. And you can see in a fairly short time, after 300 minutes, you've pretty much maxed out on the number of cells you're going to have. However, there's something slightly peculiar if you look at this. If you look at this line, it's kind of wavy, right? You'd think it would keep going out, right, or it would just go straight down. But in fact, no. You can detect that there are times when it actually, the number of cell types decreases, right? So this comes back to those funny button-shaped structures we were looking at earlier. It turns out that one of the basic features of worm embryogenesis, and this is true of all embryogenesis, of all development, of all species, is that of the approximately 1,000 cells that are generated during embryogenesis, 131 of them undergo what's called programmed cell death. That's called apoptosis, is the technical term for it. So this is one of the things that John Solston sketched out. This is one particular cell type. This is like a great-great-great-great-great-granddaughter of the first cell, and it's what it gives rise to. And wherever there's an X, that's where one of these cells rounds up. It essentially, they used to call it, cell suicide or whatever, and other cells that are surrounding it engulf it. So it's a very efficient process. All the materials are reused. Right? Now, you might think that's kind of a waste. Why would you bother to do that? That's a reasonable question. Now, in the case of a worm, it turns out you can have a mutant worm that doesn't perform apoptosis. But you cannot have a mutant mouse or a mutant human. Okay? For among other things, the reason our hands and feet don't look like that of a duck is because the skin cells, the cells in between, underwent apoptosis. Okay? And it's incredibly essential for brain development. All right? If we don't actually remove neurons that are not productive, that we don't need, we end up with severe deficits. All right? So this phenomenon of apoptosis was not quite discovered in worms. I just want to show you how important the worm field has been to this area of biology. And I should say apoptosis, well, you'll see when I show this next thing. It has amazing importance to biomedical research. All right? So cancer cells, among other things, one of the reasons they don't die is because they evade the normal signals that would trigger apoptosis. So when I did a search term just a couple days ago for apoptosis or programmed cell death, two common terms that are used, I got 2,777, 683 papers that have been published on this subject. That's a tremendous number of papers. You can see just looking at them, this one has to do with birth defects. This one has to do with glioma. That's a cancer of the brain and spinal cord. So a lot of them are biomedically relevant publications. If we go back all the way, we have to go to 13,885 pages. There's 20 publications per page. If we go all the way back, and I look at what the first publication was entered into Medline with these terms, and the phenomenon was actually discovered a little bit before that, but they didn't use these terms. But the idea is that it was in 1965, year after I was born. That was the very first paper where they used this term programmed cell death. 11 years passed, and in that time, 31 papers were published. The 33rd paper was by this guy, John Solston, who sat at the microscope, who discovered that apoptosis was a normal part of worm development. So that's in 1976. Then in 1986, 10 years later, we've had less than 200 papers published. And then a graduate student working in a guy named Bob Horowitz's lab. Bob Horowitz is still at MIT. Ron Ellis was his student, very nice guy, stayed in the worm field. He published a cell paper called The Genetic Control of Programmed Cell Death in the Nematode C. elegans. After that, the remainder of these papers were published. And I can tell you that a lot of the reasons that these papers ever got published is because the genetic program, the genes that they figured out were controlling apoptosis in a worm, turned out to be the exact same genes that were controlling apoptosis in a human, that had all this human biological relevance, let alone biomedical relevance as well. So this is just one example of many that have occurred, not just in worms, but in flies and in yeast, zebrafish, other organisms that are being used to really blow up these areas, just figure things out in a way that you could have never figured out if you had just stuck with mammalian biology. Okay, so this should answer the following question. So why do you use simple organisms to study human biology and disease? So one is that the underlying biology of all organisms is universal. What can be learned from one organism can often be applied to all organisms. And simple organisms are far easier to study experimentally than complex ones. So I tell students in one of my genetics classes, you could be, if you were an evil scientist, right, and you had no compunction, no ethical constraints on you, right, and you wanted to do experiments on humans, the problem isn't that you're evil, it's just you're stupid. Because how many generations are you going to get out of doing genetic experiments on humans during your career? 2, 3, maybe 4 if you live a really long time, right? We get a generation out of the worms every 3.5 days. Flies are about 2 weeks. Mice are, what are they, 30, 60 days? I can't remember. Okay, they're longer. Okay, and I have to say this too. The reason we share so many genes with other organisms and why those genes have very similar functions is a very straightforward explanation. That's it. End of discussion, period, exclamation point. And my point is, my point here really is just to say that we all care about doing things like expanding our knowledge and curing diseases and improving the quality of life. Those are really noble goals. And we can achieve those goals with science, but not with pseudoscience. And we have to teach the right stuff and come to grips with it. And that's the reason. Okay. That's the reason for my field. So I want to tell you a little bit about how we use the organism to figure out these pathways and networks that end up being the same, more or less, in humans. So I have to tell you there's something called the central paradigm. It's a fancy term for a fairly straightforward thing. It's how we make stuff in cells, all cells, from bacteria up to humans. As you all know, there's something called DNA, which is made up of deoxyribonucleotides. And DNA is made, after the DNA step, something called RNA is made, that's ribonucleotides, and then after that, something called proteins are made. And proteins are made up of, they're quite different than DNA and RNA, they're made up of amino acids. So proteins, we make about, well, give or take, 30,000 or so humans. We have about 20 to 25,000 genes. And the proteins are what do pretty much everything. There's lipids, there's other stuff, but the proteins are the machines, they're the structural components. Proteins are it. And really what the central paradigm comes down to is this. DNA, that's your genome. That's what's in the nucleus. That's the master cookbook. That has the recipe for everything, every protein that's ever going to be made. The RNA is basically a working copy. It's sort of like a photocopy of the DNA. It takes it out of the nucleus, puts it into the cytoplasm, because this is sort of the sacred book, and you don't want to remove that from the nucleus, and this gives a lot more flexibility. And then the final product, the thing you really needed, was the protein. Every cell makes a different complement of proteins. A liver cell will express different genes and therefore make different proteins than a lung cell, etc. So the key to understanding genetic disease, a mistake in the cookbook is going to get propagated right down the line to a faulty protein product. So here's your mutation in the DNA. That changes a codon within a gene. That changes the amino acids that the protein is making. So it folds in a different way. Maybe it has less than normal function. Maybe it has too much function. Maybe it's acquired in some sort of entirely different function. But this is the problem. Now, we all differ somewhat in our DNA, and we differ somewhat in our genes, but we're more or less all the same. When diseases occur, most diseases are in fact have a strong genetic basis. And this is what's happening. It can be hereditary. You can get it from your parents, and so be predisposed, or you can pick it up randomly because we have a lot of cells, and over time, we're going to get mutations. There's no way around it, even though we're very good at fixing them. So the approach that I use, the approach that a lot of people use, including Dustin, we sort of take this central paradigm and we sort of speed things up a little bit to our benefit. So the idea is that a researcher decides to study a biological process in a suitable organism. So if you were interested in studying apoptosis, you might say, okay, that's a good organism. It's that simple. Then what you do is you randomly mutate the organism. In this case, what Horvitz's lab did it with was worms, or C. elegans. And you look for mutant animals that would display what we call the desired phenotype. That is what they look like. So in the case of apoptosis, if you wanted to understand how apoptosis was controlled at the molecular and cellular level, you might look for mutants that don't do any apoptosis, right? Or you might look for mutants that do too much apoptosis, or maybe something in between where the wrong cells are undergoing apoptosis, or only a subset. So there's a whole spectrum of things you could look for. You might be interested in one or multiple categories. The key then is you use some techniques, experimental techniques, to actually identify the mutated gene within the organism. So worms have about 17,000 genes. There's a lot of base pairs. So finding the mutation is kind of the needle in the giant haystack. But there have been ways to do that for many years. And now with whole genome sequencing, right, it can be done in a matter of weeks. So the point is you go from this phenotype, here's a worm that fails to undergo any apoptosis, to potentially within just a few months or weeks, this is the gene, right? And you do that with lots of different mutants. And suddenly you have a whole collection. And then you figure out how they sort of fit together like pieces of a puzzle. So this is called forward genetics. One thing about it that's really important is that it's non-biased. I don't have to have a hypothesis about how this process works. I just have to know that the organism does the process. And then I let the organism tell you how it works. Okay? So by doing this, you start out, you get one mutant, right? And you find that gene A. And then you do some more genetics. There's various kinds of genetics you can do, but it's all the same principle. And you get this pathway, and you do some more, and you do some more, and you end up building up these big, complicated networks that sort of give everyone headaches when they see them. But again, what often turns out is the genes and the relationships between the genes, maybe they're activators, maybe they're inhibitors, they turn out often to be the same from a worm to a human. Biology likes to invent something and then reuse it, reuse it, reuse it. Okay, so a bit more about worms. So they're a free-living soil nematode found around the world. They have not been used as a model organism forever. They were really pioneered by a guy named Sidney Brenner in the 1970s. It was the first organism to have its complete genome sequenced. That was a massive amount of work. Took many years in the 90s. Now, again, you can do that sort of thing in just days. 3 Nobel Prizes have been given out to researchers in C. elegans in the last 12 or so years. So in 2002, this guy, Bob Horvitz, the guy who sat at the microscope and the guy who started the field, got this essentially for the discovery of apoptosis. And then these guys, Andy Fire and Craig Mello, got it not too long ago for the discovery of a phenomenon called RNAi that turns out to be a conserved process and that has really revolutionized the way people do science in all systems in biology. And then in 2008, Marty Chalfie and these other guys got it for the discovery and use of something called green fluorescent protein, which is a protein that comes from a jellyfish, which is another really obscure organism. It's the kind of thing, people could have been working here at MDBIL, 30, 40 years ago on jellyfish, and you might say, well, why do you work on that? You don't know where the next discovery is going to come from or what's going to be important. So they figured out how to make cells glow. They could attach this fluorescent module, essentially, to different proteins, and it would basically faithfully be expressed whenever that gene was being expressed. And this has totally revolutionized the field of cell biology, as well as developmental biology and all related fields in truth. Okay, so right now there's probably about 300 labs that do what me and Dustin do all over the world. There's lots in Japan and Europe. A lot of them are at medical schools, so it's used worldwide for biomedical research. Okay, so what I want to sort of close with, I might be going longer than I was supposed to, but is this okay? All right, not driving anyone crazy yet. This is where my lab is. It's on a mountaintop. But this is beautiful. I don't know what to say. This is actually pretty close to where I live. This is about 45 minutes close to the summit of Medicine Bow. You can vouch for that. So the example from my own work is one of these things where we were working on something, we couldn't have told you exactly why we were working on it, other than we wanted to do good science and figure things out. And then it sort of comes around to something that, again, seems to be biomedically relevant. And maybe we can learn something. Okay, so let me just give you a brief tutorial on worm embryogenesis. So it starts out as this sort of blob, and it starts to take shape. And as it's taking shape, the organs inside it are also taking shape. And there's an organ called the foregut or the pharynx. That's what's shown here and here and here. That gets sort of stretched out during embryogenesis. The whole worm is, in fact, getting stretched out. And you end up with a hatchling larvae that looks like this, that has this organ here that brings in food. Usually bacteria is what we feed it, and it pumps it into the intestine where it gets digested. So this is what it looks like in a wild type. This is a wild type at what we call the 1.5-fold stage. This is the mouth. This is where the pharynx is. And if you look, you can see there's this little indentation there, right? And I've blown it up so you can see it a little bit better here. That has a name that was given to it years ago called the sensory depression. There's a lot of nerve endings that end up there later on. No one really knew what the significance of the sensory depression was. It never interested anybody. There were really never any mutants in it that made anyone think twice about it. It was just this thing that was there. So we started working on these mutants, and it's a little bit more complicated. In this case, we only see the phenotype when 2 genes are mutated, all right? It doesn't matter what those genes are, but the phenotype was kind of interesting because instead of this sort of subtle little thing that kind of goes away later on, it was this pretty startlingly large ingression that we said looks a little bit like a keyhole. It gets bigger and bigger during embryogenesis. And then what happens is the whole pharynx ends up being pulled back, and this animal hatches, but it can't eat. All right? It's like its whole head has been sort of sunken in. It's like you pulled on its throat, and everything kind of sort of collapsed around it. So we were working on this, and we came up with a theory for what was going on at sort of an embryological level. We thought, you know, maybe what happens is the pharynx is like a spring inside a tube. The outside of the tube is kind of like the skin of the worm, right? And the pharynx is like this spring. It's getting elongated, but it has some resistance, right? Like a spring would if you pulled it apart. Now, in wild type, the spring gets elongated, and usually there's no consequence to that. But in these mutants, what we thought is maybe there's some inherent weakness here, right at the mouth, where normally it wouldn't have any give. But for whatever reason, because of these mutations, it's causing this ingression, and it's sort of pulling it back. It'd be like, my best analogy really is if you put a rubber band inside a tubular balloon and you attached it to the end, and you started to blow up the balloon. As you're blowing up the balloon, initially there's no real tension on the rubber band, but the farther you get out, the more the rubber band's getting stretched. It's going to start to cause the tip of the balloon to sort of ingress and pull back in, and the farther out it goes, the more severe it's going to be, sort of like our phenotype. So that was kind of our general vague idea about what was going on at a gross level. And we did a bunch of stuff, molecular stuff mainly, and what we figured out is that one of these genes, MEC8, regulates something called the splicing of a protein called fibrillin. So one of the things that happens when DNA goes to RNA is initially the RNA is made, but it's not made usually in one contiguous piece. It's sort of interrupted, and then it has to be spliced together. The pieces that are relevant have to be put together. The pieces that aren't relevant to the protein get thrown out. And MEC8 was needed for the splicing of this protein. This is what the worm protein looks like. This is what the human protein looks like. These various domains don't matter, but you can see they're similar. And it turned out what got us really interested was that mutations in human fibrillin cause Marfan syndrome. That is the cause of Marfan syndrome. Okay? So we looked, this is using, for example, fluorescent proteins like Marty Chalfie discovered. This is GFP, and this is a variant of it, actually a different protein called M-cherry that fluoresces red. And we looked to see where this fibrillin protein was normally expressed. It's normally expressed around the outside of the embryo in the epidermis, and it gets secreted. It goes, you can see it goes all the way around the outside of the embryo, right? So our hypothesis was that fibrillin is contributing to the strength and integrity of the outside of the worm. It's what was preventing the outside from getting pulled to the inside. So this is what it actually looks like. This was work done by Jim Priest quite some years ago. But what he noticed just doing electron micrographs of embryos was that they start out, you could sort of see these folds where there are individual cells, but later on, it's all smooth. And the smoothness is what he called the sheath. And the sheath is where FBN is going. So we think that this protein in worms is an important component of the sheath. And so what we think is that the sheath on the outside is really important for preventing mechanical deformation caused by intrinsic forces that normally happen during embryogenesis. Because the sheath is there, the worms don't normally become deformed. But when the sheath is weakened, they do become deformed. So this is an example. There are various squeezing forces, and this is the pharyngeal pulling force going in right here. This is sort of the tube model again. So we get this phenotype. Or if we really weaken the sheath, there are some other constricting forces that really lead to this rippling within the epidermis. And essentially what's happening is the epidermal cells are becoming hyperextendable or hyperelastic. Well, that turns out to be one of the clinical manifestations, not only of Marfan syndrome, but a whole bunch of related diseases. Fibrillin 1, fibrillin 2, collagens, elastin. And actually, the skin is the least of the problems because I think you can live with elastic skin. What you can't live with so well are things like, elastic components of the heart, etc. Other vascular tissues lead to real problems. So this hyperelasticity is something that we're seeing in the worm, and it's also something that is noted in the human disease. So here's some thoughts of sort of where I think this project could go, and again, why we do these studies, because we don't know where they're going. And I still don't know where it's going, right? But one thing I know from reading the literature is that the severity of Marfan syndrome varies dramatically between individuals, including individual siblings that have the identical alleles, right? They have the exact same mutation. One shows a very severe form of the disease. Another one shows a very mild form. Why is that? It could be partially environment, but mostly it's probably because all the other genes that are present are considered to be genetic modifiers. So depending on what else is present, that same mutation might cause a severe effect or a relatively minor effect. This is also true in mice. If you do a knockout of the fibrillin gene in mice, some mice show a severe phenotype, some show a mild phenotype, but they're different strain backgrounds. And so it's thought that the background is doing this. So essentially, what we've been doing in our lab is finding genetic modifiers for the fibrillin-like protein. No one knows, despite the fact that this is known, no one has identified a single genetic modifier of Marfan syndrome in a human. But we've got a bunch of them in the worm. So we can hypothesize that maybe the same things that are modifying the phenotype in the worm are modifying the disease in a human. So can worms be used? We hope so. The other final thing I'll say is that FBN1 mutations in many Marfan's patients occur at sites that affect the mRNA splicing or processing. No one knows what does the splicing of fibrillin, the fibrillin gene in a human. Well, do FBN1 splicing factors contribute to the disease? So do splicing factors contribute to the severity of the disease? That's something we wonder about. And we've identified this protein called MEK8 that has a perfect human component, or ortholog, which we know regulates fibrillin splicing in the worm. Perhaps it does so in the human, and maybe that has a clinical consequence. So with that said, I will close. So that's sort of my own, there's lots of examples. We all have examples like this where we've been doing things. We couldn't tell you exactly why in all cases we're doing them. In some cases we can, but usually when we can, it ends up being something different. And in the cases where we're not, it ends up going in the direction you'd hope. So I've got some good people in my group: John Yoakum, Melissa Kelly, Alexandra Kuzmanov, Vlad Laztek, and Evgenia Karina, various collaborators. And so I'll close with that, and that sort of hopefully gives you some idea about why we would use such an obscure thing as a C. elegans to study anything related to human biological and diseases. So thanks.
Thank you, David. We have plenty of time for questions, so go ahead. Anything you want to know about the worm or how we use genetics?
Do you think the splicing factors are mutated themselves?
So the idea could be that so the question was, could Could there be mutations in splicing factors that either lead to the disease or affect the severity of the disease? Is that sort of your question? Yeah, I think that could be the case. But no one knows what they are. We don't know if this will directly translate or if they are in fact important in the human disease. But that would be the idea. Yeah. Sure.
Well, how do you make the next step to that?
That's a good question. So the question is, this is a great question actually, and I used to have a slide sort of like to address that sort of question. How do we make the next step with that? So what we do, I sort of, a man's got to know his limitations, right? Like Clint Eastwood said. So I believe, I know I'm pretty good at doing worm stuff in cell biology as relevant to the worm. And occasionally we'll venture into mammalian tissue culture and other things. But frankly, there are other people who do a much better job with that or just have it set up. It's easier for them. So I'm going to figure out everything I can. I'll play to my strengths as they are and figure out what I can in that situation. And then, very importantly, you publish papers. That's how you do it, right? It gets out there. Immediately, everyone can see what you've done. And then what happens, I mean, the reason those 277,000 papers is that other people saw Bob Horvitz's papers. And they said, man, we've got to check if this is going on in mice or in whatever cell line, and so it gets passed up the line. I mean, if you really think about the true chain, right, what you ultimately care about is something that would affect clinical outcomes or prediction or, diagnostics, something like that. So again, this is quite a few steps removed from that, but that is how it works. It's got to start somewhere. So it's kind of basic science, foundational stuff that I do, that Dustin does, that a lot of us do. Yeah.
About 20 years ago, one of our colleagues at Columbia University got well known and indeed got the Nobel Prize for doing some very powerful things with this same worm. Could you give us a little...
Marty Chalfie?
Excuse me?
Marty Chalfie?
And some colleagues.
Okay.
The guy who got the Nobel Prize in 1998 was one of our other people. But
Right.
could you trace for us what it is about this remarkable worm and the things that you've been able to describe that got the attention of the people who were looking at the connection? And genetics got invented, by the way, after I went to medical school and a lot of other people did. And boy, has it changed the way we do things. But could you trace for us why this was so exciting that it got up to the people in the Nobel Prize thinking maybe this is something they wanted to reward?
Okay, like the Nobel Committee, why they... So I think, all right. So I think the reason, for example, the reason the discovery of apoptosis got the Nobel Prize is the 277,000 papers. And, I don't know too much about how Nobel committees work, from certainly from the outside or inside, right? But, I mean, I think most people working in our field would have said, absolutely, this was deserving of a prize. That took years before they got that prize. From their initial paper, you're absolutely right, it was 1998, and so that original paper was what, 80? Actually, yeah, it was a good distance. The other Nobel Prizes came more quickly. Part of that is the pace of science. Science pace just keeps picking up and picking up. And the other thing is that the two other Nobel Prizes had very strong methodological components. So the RNAi discovery, it wasn't just a biological discovery. The other half of it, or maybe more than half really, was a research tool. It allowed everyone in every system to knock down any gene of their choosing at any time, more or less, using that type of approach, at least within certain constraints. And so that really revolutionized within just a couple years the way people were doing experiments. The same thing with the GFP. That was this guy Marty Chalfie at Columbia. The way, and again, within just a few years, everybody was doing it. All right, the news spreads quickly. You're left in the dust if you don't adopt these new techniques. And why would you? They're in some cases very easy, and they're certainly powerful. The people who were working on this stuff, most of the early people, like Horvitz and some of the others, they didn't start out as worm people. They started out studying a virus that infects bacterium, the bacteriophage, for which Nobel Prizes have also been given out. And they decided at some point in the 70s, a group of them, that, we think our field has played out a little bit. It's more limited, what we're going to get out of it by putting more into it, there's going to be diminishing returns. And so they did something fairly brave, right? Because they were still doing very well in that field at the time. They said, we're going to drop this. We're going to drop this component of our research. We're going to strike out and start working on C. elegans, which no one had heard of before the 70s. So there has been genetics for quite a few years. There was Ed Lewis and some others were doing genetics a long time ago, but what has changed is the molecular genetics part. You could do genetics and get interesting mutants and say, wow, that's a really interesting mutant. And people could say, yeah, who cares, right? So it's an interesting fly mutant, whatever. It wasn't until they could clone the genes and say, well, this is the gene that caused that effect in the fly, and look, there's a human version of it, and when you mutate that in a mouse, it causes something that looks just like it, and we sequence these patients, and they have this mutation in that gene. So that was a bit of a rambling answer, but for somewhat different reasons, they all got the Nobel Prize. And there could be a fourth one coming out within a few years, too, for a different discovery as well.
It took off so fast.
Yes, everything moves incredibly quickly. And you have to be ready to move with those changes. Yeah. Sorry.
I guess 300 labs working on this, probably 1 million papers on all these other things besides apoptosis.
Right.
And I'm thinking Cynthia Kenyon and life extension, 5 times age normal. Now,
Right.
what I'm going to ask you is, besides Cynthia Kenyon, what other great applications or things have been discovered through research on C. elegans?
Okay, so most of the work that's been done on C. elegans has been done on genes, not all of it, but most of it's been done figuring out the basic functions of genes that have conserved counterparts in humans. So you could say that everything that's been done by the whole field, not quite everything, but a lot of what's been done, has shed considerable light on functions of human genes that no one had any clue about before this was done. All right, so that's a general contribution. The 3 Nobel Prizes are probably legitimately the greatest highlights of what the field has accomplished. But, I just think in general, if you add up the total accomplishment, it's all the papers that didn't lead to the Nobel Prize, for sure. They vastly outweigh the few that did, just in moving the entire field of biology forward in terms of understanding what genes, proteins physically, functionally do at a molecular and cellular level. It's interesting about the aging question that who Cynthia Kenyon is and whatnot. So, this is an area that is actually a fairly controversial area, I would have to say, in the worm field. We're not all great believers in the aging field for various reasons. So there are mutants in C. elegans. People love this stuff, and it makes a lot of splashes in the New York Times and other high-profile newspapers and magazines. You can mutate genes in a worm, and the worm lives 3 times as long as a normal worm. So I didn't say anything about the lifespan of a worm. It lives normally about 17 days. Depends on the temperature, but in the laboratory. We don't really know quite how long it lives out in the real world, but it lives about 17 days. It turns out you can knock down a gene. You can get a mutant in a gene, several different genes, that cause the worm to live 40 days. There's a fly gene called Methuselah. So we have this really proper English guy who ran the worm nomenclature, and you had to get an approval. He was very stuffy, and if he thought you were being a little bit too, just flashy or something, he would shut you down. We have really boring 3-letter names followed by a number. The fly field has no such constraints, so they get to name their genes all sorts of crazy things that actually get pretty confusing as well. So I think flies live normally a couple weeks, and Methuselah flies live, that's like, a 6 or 7 times lifespan extension. So what some of these people have said, well, man, there are human versions of these genes. All we have to do is knock down this gene in a human, and we'll all live 300 years. I would dispute that that's going to happen. And I would also, for a lot of reasons actually, but for among other reasons, here's why here's I think a pretty convincing argument why it can't be that simple in a human, or it probably isn't, because we've never known a person who's lived 300 years, right? If it's we're a walking test tube. The whole human population is, right? So we all have different mutations. In fact, if we have really deleterious ones, we never get here, right? So if it was as simple as knocking out one gene or inhibiting the expression of one protein, I think there would be random people, 1 out of a million people, or 1 out of whatever the mutation rate is, that would be that old. Now, I've come up with some caveats to that argument, but anyway, yeah, the aging field has a lot of hype, but I think, I would be cautious of that in particular. Yeah. Yes.
Would you talk a little bit more about that programmed cell death?
Sure.
What you've learned from it, how it applies to humans, maybe, or not?
Right. So the basic pathway in C. elegans, I'm trying to avoid the use of gene names because they're kind of
A lot
pointless.
of smart people here.
Right. Definitely, but it's like so one of the things that was discovered in worms is a lot of the endpoint regulators of apoptosis, caspases. These are proteins that chop up the cell and essentially prepare it for death. They're proteases. Those are several of there's a number of those in C. elegans, and those were discovered at the terminus. Upstream, there are other genes that basically tell these caspases to become activated. Once the caspases are activated, it sort of reaches a tipping point, and it's like the cell is now going to die, right? Plus, there's the engulfment genes and various other things that turn out to be conserved as well. There's one good example. There's a gene in C. elegans called SED9. So Horowitz isolated a gain-of-function mutation in SED9. And the gain-of-function mutation prevented cell death. So that was one of the first findings that turns out, even though they discovered it in worms, it's not essential for viability. There are some consequences, but worms can live with the extra 131 cells. It actually affects the germline more in the end, but that's okay. So this gene SED9 has a correlate in humans called BCL2, and the BC stands for Burkitt cell lymphoma, the BCL. So in this particular, I don't want to get the details wrong, but essentially there's an amplification of this gene, or there's a translocation so that this BCL2 protein is made in much higher abundance in this particular lymphoma than in a normal white blood cell. And what that does is it prevents those cells from undergoing, these are progenitor cells, but it prevents them from undergoing cell death when they normally should, so they overpopulate, right? And that's one of the hallmarks of cancer is hyperproliferation. So the other side of hyperproliferation, there's the sort of the cell growth and division side. There's also the not dying side. We're really good at killing cells that we don't need. We maintain what we call homeostasis, right? So there's constant proliferation, which we need throughout our entire lives, but there's also balancing that. We have to do apoptosis. For example, cancer is one thing, but your immune system's another. The reason your immune system doesn't destroy you, right, is because the immune cells that produce the antibodies that are self-recognizing get weeded out through a process of apoptosis, right? So you can't have a mouse or a human that doesn't do apoptosis because we never make it very far. You can have a worm. They have a very primitive immune system, so that's not a problem in that case. And they don't live long enough to truly get cancer. 2 weeks is too fast, all right? So those are some of the genes or aspects of apoptosis that is translated. But there's literally not a process you can look at where apoptosis doesn't play some role. Certainly no developmental process. Yeah?
What about, this is again one of these disruptive longevity questions, the calorie restriction.
Caloric restriction, yeah.
Does research on the worm shed any light on how that might or might not work?
Right. So there's a guy in Boulder, Colorado who I knew when I was there. His name is Tom Johnson, who did some work on this. So what is known is that caloric restriction in a rat will increase its lifespan quite a bit. And it turns out caloric restriction, when applied in a certain way, can also increase the worm lifespan a lot. And some of the genes that are required for lifespan extension and caloric restriction have been discovered in the worm. In fact, one was a gene I was working on. This is a funny thing. I was working on this gene in a somewhat boring context, but I made some, I do a lot of that, but I made some connection. And I figured out that several proteins were interacting and doing something, but I didn't know the meaning of certain ones. I was interested in some that they made sense to me. And then this guy, Andrew Dillon in San Diego, published a really nice Nature paper, which is a lot better than the papers I was publishing in the same area, where he actually discovered one of the genes that was required specifically for lifespan extension under caloric restriction. And I got a nice email from him. It said, everything you said was true, you know, in my paper, right? And that for a scientist, you know, we can't control where it's going. But especially if another scientist said, you know, what you said, it wasn't crap, you know, we're really happy that someone noticed, you know, and that it wasn't, in fact. We're relieved, in fact. But so I don't know, you know, it's funny, Cynthia Kenyon, I've had dinner with her, and she practices caloric restriction. She ate like 2 lettuce leaves. It was amazing. It's like free food. So I go back to my undergraduate, like free food. I mean, what more do you need to tell me? But yeah, so I don't know a lot about. I mean, we all know we're supposed to eat healthy, right? But I don't know how much. You know, there's so many indirect effects. In the worm, it's very clear and it's direct, right? There really is, and in rats, apparently, it's quite clear too. I can't answer your question much more than that, though. Yeah.
Yeah, coming completely from outside science, I wonder if it's kind of pure research like this with no miracle drug at the end.
Right.
How does that get funded?
It gets funded by you. You fund it. You may not. So part of your tax dollar goes to the National Institutes of Health.
I just kind of hear certain people saying, well, I think tax money goes for studying worms that are only this big.
Absolutely, and that's very true. So that is something that happens sometimes. I think was it Dave Barry who wrote there was an article. So there's a guy in Seattle, Jim Thomas. Yeah. He works on worm pooping, or he used to. And what he figured out was that worms poop every 32 seconds. On the money. They are regular. And what he was really interested wasn't so much in worms pooping, but the fact that there was a neuronal circuit that could perfectly time 32 seconds in between contractions. And understanding what was the molecular basis, because there's all sorts of correlates in other systems, right? Sort of these little neuronal circuits that are timed. However, so it was actually a very valid thing to study at a biological level. It was just the outcome that was sort of embarrassing. But Dave Barry got I think it was Dave Barry who wrote a comical article, right, about there's a guy who gets $1 million from the NIH to study nematodes pooping, right? So to most people seeing that, they'd be like, what is going on here? I mean, that's my tax money. Why are they wasting this money? You know, it's I'm not saying that some bad research doesn't get funded by the NIH, you know, but a lot of our government wastes monies in way worse ways than it ever could imagine, right? So it's mostly NIH. You know, there's some other great organizations like, well, there's NSF too, national, but that's your tax dollars. But there's private organizations. I'm sure a lot of you have had to do with American Heart Association, American Cancer Society. The ACS is what gave me my first grant. And it was a good one, and I've always appreciated it. So there's private foundations as well. Oh, sorry.
Last time we had,
Go ahead. Yeah.
last time we had something on regeneration. Can you cut one of your worms in half and get 2 regenerations?
Our worms are absolutely pathetic at regeneration. So like the great thing, flatworms, planaria. All right. There's not that many people still working on planaria, but planaria is, you know, these flatworms that live in fresh water, like pond water sorts of things. And someone figured out in the 1800s that you can cut them up into exactly 264 pieces. If you do it 265, it doesn't work. It's literally something as crazy as that. But you can cut them up and they completely regenerate. And that's because they're full of stem cells. They have stem cells distributed throughout their whole body. The worms that we work on, part of the problem with that hardwired lineage is it's hardwired. You differentiate and that's it. There's no going back. So there's no regeneration capacity whatsoever. Now, that said, people do study stem cell aspects in worms. So there are cells that hang out for a while and then proliferate. And also the germline is sort of the ultimate stem cell, right? So worms have a germline. So certainly things have been learned, some important things that have translated well into other systems by studying how worm germ cells do their thing at a regenerative level. Yeah.
I'm bad at math, but you said 277,000 and some odd pages.
Yeah, exactly.
Okay, well, that would be more than 800 years.
Yeah.
If you read each one paper a day for 800 years.
Yeah.
So how in the world are you going to, I mean, how can you keep up and how do you know that what you're doing is relevant if it hadn't already been done or something?
That is a great question, and well, sort of welcome to my nightmare. So I know my area pretty well, and I can do searches in fairly smart ways to let me find things, or I have them done automatically for me and they give me alerts. So, but you're right, let's just say you're interested in apoptosis. So a lot of those papers, even though apoptosis crops up, if you're working on, you know, we're specialists. We work in this and this and this. And so we need to know what's going on in our own area and how it informs our own research. And we need to see, you know, we can tell who cited our work to know where it's going, et cetera. But it is a problem, and I worry about that too. I mean, the fact that what ultimately distills this knowledge into something that's actually usable to a mere mortal, right? I mean, a computer can know it, but that doesn't help us, right? But it plays out. It's not quite as bad as it sounds. But you're absolutely right in what you're saying in a general sense. But I feel like I'm pretty sure I don't miss things that are directly relevant, that if I should know about it, I probably do. And that's why I kind of stay within the worm field. I do my thing, and then, you know, other people you do have to count on other people noticing. I mean, partly you go around, you give seminars, but it's mostly just people pick it up in the literature. You don't have to scan through them. Everyone has these sort of smart searches that they do, right? And it spits out every week or every couple days, you might get an email that basically says, well, these are papers that might interest you. You kind of shuffle through them quickly, looking at the titles. Maybe you go to an abstract. Maybe only one or two of them are actually relevant that you're like, oh, I really need to read this carefully and see what happened. Most of them you can kind of push to the side. It doesn't mean no one's reading them.
Thank you. Thank you, David. We'll go ahead and end with that.
Oh, okay.